Okay, hi everyone, how are we? Today we're going to be going through the rest of mitosis and the beginning of HSC Biology. And what I'll in fact do is I'll spend this lesson going through the first inquiry question which we skimmed over, which is methods of reproduction. Now for those of you who just joined, very exciting, your first lesson with me. We're going to be running through the start of HSC Biology Module 5. Okay, so key thing for everyone, remember this is very discursive. I want you to ask as many questions as you'd like, right? My goal here is to help you out in a very discussion or informal format and make sure that number one, you understand the content, the beauty of biology, but also to make sure all of you get the mark you want. And I'm sure most of you here are interested in medicine. So as you know, it's quite a high ATAR course and I will help you on that journey. All right, now, before I start, did anyone do anything interesting in the week? Anne, did you get up to anything over the week?
Not really, but I got my senior jackets on Friday.
Nice. I remember we used to be able to put our credentials or our little nicknames in the back, which was nice. But that was many years ago. For those of you interested with medicine, I'm currently on my final couple of weeks after med school exams. So I'm doing rotations in surgery and radiology. So that's imaging. I'm doing colorectal surgery as well. And it's quite relaxed. When you don't have exams in medical school, life is much easier. But we'll talk about that and career planning during our break as well. Now, to start us off, let's start at the very beginning. I know we spoke about mitosis last lesson, and I'll quickly quiz you on that, but just to give you a little bit of a cap on where we're at. And these are my notes for those of you who are new to today's lesson. This is everything you will need to know for HSC Biology. And we will start all the way at the front. Now, students who were here last week, you should have electronic access to the notes. And I'll talk about how to get printed access to the notes in the break time. So someone just remind me for that. But those of you with the notes, I want you to go all the way up to this page here, page 22. Okay, and those of you who don't have access to these notes, completely fine, I'll give it to you at the end of the lesson. For now, just use your books. Okay, good. Now, let's quickly recap what we learned last week, and I'm going to go through this again, so not to worry. We learned about mitosis and DNA structure. So I'm going to start with, Ruthania. Ruthania, what does DNA stand for, and where is it present?
DNA stands for deoxyribonucleic acid.
Okay, cool. Now, I don't care if you guys can just learn and say it back to me, but you really need to understand why it's called this fancy term here, right? So to answer that question, Sarah, what are the 3 repeating components of DNA, which is our double helix? What did I say kept on repeating itself in its structure?
A and T, C and G.
Okay, so that's one component of the repeating unit of DNA. We have the base. What's connected to the base?
So it's the...
Yeah, anyone? Sarah?
Sugar and phosphate.
Very good, right? Base, sugar, phosphate, right? So your sugar, phosphate, base, and it keeps on repeating, right? Next to this, you're going to have another sugar. You're going to have another phosphate, and you'll have another base, right? So you can see the repeating unit is this 3-unit structure. What is the name of this structure, Ed? What do we call this?
I forgot.
Let's go to Matthew. What do you call this three-component structure?
I don't know this either.
Okay, right. I recall we briefly went through this, right? Which is it's called the nucleotide, the basic repeating unit of DNA. So the nucleic acid comes from the fact that DNA is made out of a base, a sugar, and a phosphate, right? That represents the nucleic acid part of it. When you combine all of these and you measure the pH is less than 7, hence why DNA is actually an acid, right? Now, why is it called deoxyribonucleic acid? Who can tell me that? Let's go to Anne.
Is it because the sugar was deoxyribose?
Yeah, right. So there's many sugars out there. There's sucrose, there's fructose, there's maltose. There's also ribose sugar, and when you take one oxygen atom off the ribose, it's called deoxyribose. So now, hopefully you'll understand the entire unit of this name. Now, I want to make this very, very clear. None of you are going to remember this name at all if you don't say it, right? So the power of most of you being on mute right now is when I teach you a new word, if you don't say it and include it into your vocab, you're going to forget it within the end of the day. I can guarantee that, right? So repeat the terms that are new and scientific that we come across in the lesson, and they'll stick. Okay, good. So that was a very brief recap about DNA. Now, let's talk briefly about mitosis. What are the 3 reasons why we need mitosis? So mitosis is when we divide to make 2 genetically identical daughter cells, right? So we copy the DNA, we split the cells into 2. Now, Ed, why would an organism want to undergo mitosis? What are the 3 reasons?
For growth and repair, and then
Good.
reproduction and some.
Not really reproduction, right? Because you don't just copy an offspring organism. It's much more complicated than that, and that's what we're going to talk about for Module 5, HSC Biology. But mitosis is required for growth. It's required for repair. Who can tell me what the last reason for mitosis is?
Maintenance?
Very good, right? So we need it for growth because you were originally one cell and you multiplied now and you're all multicellular, billion-celled creatures. Mitosis is needed for growth, right? If you cut your skin, mitosis is needed to replicate skin cells to cover the gap over the paper cut, right? And that happens without you even thinking. And the final reason is maintenance, right? Interesting fact, your skeleton replaces itself every 10 years. Right, the entire 40, 50 kilos of skeleton that you're made out of every 10 years replaces itself. And the way it does that is largely by mitosis as well. Very good. So that is mitosis. Now, very briefly, last thing I'm going to talk about before we jump again to the start of Mod 5 heredity is the cell cycle. It's kind of like a life cycle, right? All of you have a teenage part of your life cycle, which you're at right now. You're going to have a young adult life cycle, which is where I'm at. Then you're going to have an old adult, elderly, and then end of life. In the exact same way, a cell propagates through different phases. What are the names of these different phases within mitosis, everyone? Has a little acronym, and you would have been introduced to it last lesson. Bethania, any ideas? What is that acronym I'm talking about?
IPMAT?
Very good, right? So IPMAT, who can tell me what it stands for? IPMAT. Any ideas? Let's go to Ed. What do you think, Ed? What does IPMAT stand for?
Interphase, prophase, metaphase, anaphase, and telophase.
Okay. Now, I understand for a lot of you, this means a whole lot of nothing, right? Just a bunch of fancy looking words. So do not fret. I'm going to teach you all about this. But I just want to highlight something very important now, right? When we talk about the cell cycle, a cell can either be doing nothing and happy, making proteins and doing its job, or it can start the process of dividing, which is known as a cell cycle. Now, the majority of the cell cycle, so this entire part of the life cycle of a cell, as you can see here, is spent in a preparation phase known as interphase. This is when the cell is building the nutrients, it's getting ready to split into 2, because it's a very, very complex task. That is what interphase is. So the majority of the life of a cell is stored within the interphase stage. Now, we spoke about DNA, and you all know when a cell splits into 2, everything within it needs to split into 2 as well, including the DNA. Can anyone tell me which phase of mitosis, is it E, I, P, M, A, or T, does DNA replicate?
Interphase.
Very good, right? So in that initial preparation stage, interphase, that is when your DNA is going to double. DNA replicates. Okay, and the rest of the cell, this tiny remaining fraction, is spent in prophase, metaphase, anaphase, and telophase. And that is the rest of the division. So we're going to talk about that later, but the PMAT phase represents when that cell physically will divide into two daughter cells. Okay, so that was a very quick recap on what we learned last lesson. And all of you who weren't here, you'll get access to the recording, so not to worry. Let's talk a bit about heredity. So just very quickly recap, why is the module called heredity? What are you all going to learn? Matthew, what do you learn in this module? Because you need to have a bit of a snapshot before you jump into things, right? What is Mod 5 heredity?
We really just talk about genetic information and gene sequence of DNA.
Good. Genetic information. The other thing I would add is you learn about genetic information, and you also learn how that genetic information is passed on, right? So the first half is going to be all about genetic information, how is it stored? Then you're going to learn how is that genetic information passed on. Now, let me ask you all, why should you care? Why does it matter for society or our population or science to learn about genetics and genetic information? This isn't something your teachers will teach. It's a very logical thing. Why do you guys think we need to care about DNA and genetics? Can you guys hit the raise hand icon? Were you born in New South Wales? The raise hand icon if you're born in Australia. Okay, good. Now, every one of you that were born in Australia, without your consent, because you're a baby, we pricked your heel for blood. We took 3 drops of blood, and we looked at that 3 drops of blood. We look at the DNA within that blood. We looked for about 33 different genetic conditions that could have killed you before you reached adulthood, right? 3% of babies will be born with one kind of genetic defect or disease. Now, if we pick it up through that genetic testing, we can treat that disease early and save lives. Now, this is just one of the billions of examples, the top one that came to my head. But that's a direct use of our understanding of genetics to identify disease early, treat children, save lives, and contribute to humanity. It all comes from our understanding of where is the genetic information stored and how do we read it. Okay? So that's just one of the multiple expansive examples of genetic disease. And I want you all to understand we're getting to a point now where by the time you're all adults, sequencing your genome, so that's all of the letters in your DNA sequence, the billions of letters that tell your story of how you live, what diseases you'll get, what you'll die from, in fact, will all be stored in a little USB stick. And eventually we'll get to a point where the doctor can plug that into a computer, read the script that is the language of your life, and tell you what diseases you'll get, at what ages you'll get them, and your most likely causes of death. Right? So society is undergoing a tech revolution, but it's also undergoing a biological revolution with health tech as well. So those of you who understand this stuff really well will be uniquely positioned to solve a lot of problems and reap a lot of the rewards as well. Okay? So hopefully you understand why you're learning this, because I don't want you all to just rote learn biology. You should understand why you're doing this. Now, we spoke about heredity. The reason I spoke about this last part is you're going to learn about this, which is finally DNA technologies. How do we use information from this section of the module to contribute to society, our understanding of human evolution, and health as well? Good. Does anyone have any questions about the module heredity? It's all about genetic information, DNA, how is it stored, how is it passed on, and what tech comes out of that knowledge. Any questions at all? All right, very good. Now, here we have inquiry question 1. Now, Rithani, can you remind the class, why is the syllabus stored as inquiry questions and dot points? What's the purpose of this?
If you treat the inquiry question as a question, you're expected to write about all the dot points.
Yeah, very good, right? So the dot points are what you go through. They're like little side quests that you go through. Once you finish all the little dot points, you can attack the boss, which is the inquiry question, right? And you should be able to write a full answer to the inquiry question that stems from the dot points. That's what I want you all to get to. So when you finish your notes, try doing a dot point answer to this inquiry question, which is how does reproduction ensure the continuity of a species? Now, I want you to pause and think about this. Before we get through the content, I need to know your baseline understanding. To read this question, and I'm going to ask one of you to answer it for me in layperson words, right? I don't care about using science and fancy terms. Just tell me how you think reproduction makes a species continue going. And for this, we need to define the question, right? What is reproduction? What does continuity of a species mean? Because once we understand that, we can answer the question, right? So, what is reproduction? Let's go with Alyssa. Alyssa, what's your understanding, just layperson understanding of reproduction? How would you explain it to a friend?
Like when offspring is produced for the continuation of a species.
Very good, right? So, okay, I like that you use some keyword offspring. Now, this is what you have to understand. Offspring doesn't necessarily mean a whole multicellular organism, right? Right now, for every skin cell all of you have 10 bacterial cells. So you're actually more bug than human. But those bacteria are constantly dividing, and they're making offspring. It's just the offspring are just one cell, right? So the term offspring, it doesn't mean a whole organism. It just means making another copy of itself, whether it be a multicellular organism like a plant, animal, or whether it be a single-celled organism like a bacteria. Very good. So creation of a new offspring. I like that. Let's go with that. Creation of new offspring. Nice. Now, what ensures continuity of a species? If I gave you a little human, a 3.5 kilo baby in your hand, and asked you, how will this baby continue the Homo sapien species? What are the two things that need to happen for this baby for it to continue the species, per se? Think hard about this one. Let me start with asking Matthew. Little baby needs to continue the Homo sapien species. What are the two things it needs to do? Or just tell me one.
It needs to be able to reproduce or reach adulthood, and needs to be able to have a different personality. A different trait that makes it different.
Yeah, look, you already nailed both of them. I was literally just looking for it needs to reach adulthood, so reproductive maturity, and it needs to have a means of reproducing, because then it will make a new offspring. And whenever you have offspring over time, you have continuation of the species. So very good, right? The two things that an organism needs is it needs to grow to reproductive maturity, and then it needs to have a mechanism of reproduction. This little flowchart is our literal answer to how reproduction ensures continuity of a species. You could write this. Therefore, you've got your answer. Now, this is a very simplistic answer. So let's delve into it in a little bit more detail. How does an organism that is originally one cell sitting in the fallopian tube and later into the uterus, how does it divide into a 3.5 kilo baby and then grow into individuals that can be multiple kilos tall in height? How does that happen? What cellular process is driving that, everyone?
Mitosis.
Very good, right? So to reach reproductive maturity, you need a huge amount of mitosis. Not just for growth. Atul, what else did we need mitosis for? So growth is just one of the reasons. What are the other ones? Growth.
Repair and maintenance.
Good. Growth, repair, and maintenance. Very good. Now, Atul, if you can hear me, question to you. We've spoken about mitosis, but how do we make this organism, person one, produce an offspring? Do you know about any cell processes that are required for this?
Meiosis.
Very good. Now, tell us a bit about meiosis. What is meiosis, and how is it different to mitosis?
I don't know a lot, but I think it's like reproduction of sex cells.
Yeah, very good, right? That's literally it. It's how you make sex cells, but then the question is, why sex cells? Why are they important? Any ideas? Let me go to Anne. Anne, what are sex cells?
So I think they're called gametes,
Very
and
good.
there's like the egg and sperm, and they are haploid cells, so they contain half the genetic chromosomes. So when they combine, that's when like a human starts to form.
Very good. You've used all the buzzwords that I was going to explain. And to the rest of the class, let me quickly explain what she mentioned. Now, when we talk about meiosis, again, right now, I understand you all know nothing about mitosis, nothing about meiosis. So we're going to fill in all those gaps later. For now, I just want you to know what those processes result in, and I'll teach you later how the process actually works. Okay, so bear with me. Mitosis, you get one cell, splits into two, genetically identical cells, right? So it's happening in your body. It happens about every 48 hours for a cell. Now, meiosis, you don't make two cells. Bethania, how many cells do you make? And are they genetically identical or different?
I'm not sure how much you make, but I think they're not identical.
Good. They're genetically varied. And to answer that question, just look at your sibling, right? They look different to you. The reason was because the cells that we made from meiosis to make your siblings are genetically varied and hence different to the cells that combine to make you, right? So meiosis makes 4 genetically varied cells. The cells have half the genetic information, okay? And it actually makes sense when you think about this. You have one cell. In cell division, DNA is going to double. Everyone agree? So if I have 1 copy of DNA, it's then going to split into 2 copies. But then when you divide it into 4 cells, 1, 2, 3, 4. If you had to split $2 between 4 friends, year 3 maths question, how much money would each person get, Anne?
So what was the first part?
If you had to split $2 between 4 friends.
50 cents.
Very good. Same way, if you had to split double genetic information between 4 cells, you have half the genetic information in each cell. Now, the way I remember this is the word half reminds me that these cells are haploid. Haploid meaning half the genetic information. Okay, very good. Now, for reproduction, I want you all to understand this. Meiosis is not reproduction. They are different things. Meiosis is a precursor to reproduction. Okay, so reproduction, if you think about organisms like us, it's a process by which you combine these cells that you already made from meiosis. So you need meiosis to happen first to make these haploid cells. They're known as gametes or sex cells. You can call them whatever you like. Does not matter. I like to call them gametes, right? So these gametes have half genetic information. When 2 of them combine, you're going to produce an organism with a full set of genetic information, right? Very good. Good. Any questions so far? All right. Now, very briefly about reproduction. Meiosis is a precursor. You make the gametes, they need to combine through reproduction. Okay? So we need meiosis, but the actual mechanism here for all of this to occur is we need a mechanism of reproduction. Okay, what I mean by that is in terrestrial organisms, it's mating, right? Mating is how you combine the 2 gametes. The gametes were made by the individuals prior to the mating process. So meiosis is needed beforehand, and then we need a mechanism of reproduction. So guess what, everyone? You have answered this inquiry question. So in summary, what I want you all to understand is reproduction is a mechanism which allows gametes to combine together and make a new organism. And that's how it ensures continuity of a species, right? And there are different forms of reproduction which we're going to talk about. But is reproduction the only thing you need for continuity of a species? Matthew explained this, but let's go to Atul. Atul, what are the other features you need for continuity of a species? Reproduction is just one of them.
Variation.
Yes? Repeat it.
Variation.
Sorry, could you repeat it one more time?
Variation.
Yes, so variation from what process? Meiosis, right? Meiosis is how you make genetically varied daughter cells, which will then combine and make varied new organisms. What's the last thing? There's one more process that is essential for continuity of a species, everyone. What is it? It's written here. I just want someone to say it. What is the last process we need? Sarah, we need a mechanism of reproduction. We need meiosis. What's the last process we need to continue a species?
Reproductive maturity.
Yep, remember, that's not a process. When an organism reaches puberty, they've reached reproductive maturity. I'm asking, what's the process that allows an organism to survive and grow till they reach that stage?
Mitosis.
Very good. So guess what, guys? You understand the three most important parts of this inquiry question. Reproduction is a mechanism to make new organisms, ensuring continuity of a species, but it's not the only thing. To have continuity of a species, you need mitosis and you need meiosis. Why do we need mitosis? For growth, repair, and to reach reproductive maturity. Why do we need meiosis? Let me put that out to you guys. Alyssa, how would you explain why do we need meiosis for continuity of a species?
Because when the gametes fuse together,
Yep.
it creates randomized so it creates a genetic variation.
Good, that's one part. But remember, if you didn't have meiosis, reproduction would not form anything, right? Because you don't have the cells that will combine to make a new organism. Reproduction is just a mechanism, right? If you don't have the gametes from meiosis, there is no reproduction. You see that in the human population. About 1 to 2% of humans are infertile. They can't make gametes. Their meiosis is not working. They can't continue a species, right? Even if they engage in reproductive methods. Hopefully you're understanding the three things you need. These are literally the triad that you need for continuity of a species. The reason I'm mentioning this is if they ever ask you a question which says evaluate the importance of reproduction in continuity of a species, your answer should say reproduction is important, but two other things are also as important. And you will list mitosis and meiosis. Okay? Very good. Any questions? All right. Now, this is where we're going to go back to Year 11 content. Now, this is an interesting Year 11 content. I can appreciate Year 11 was cool for the first half for me. And then Module 3 and 4 were also interesting, but I'm not too fussed about plants and what they do. But I think once you understand what plants do, it's much easier to understand what complex or even more cellularly complex organisms like humans do, right? So we're going to jump back to Module 1, Year 11, and I'm going to test your understanding of kingdoms and what the different kinds of cells have. Now, I want all of us to read this document. I'm not going to get someone to read it out for us, but have a quick read of 1.1. It's very long, and I'm going to ask some of you to dumb it down for us. What do we need to know to do well for this dot point? This is a very hefty dot point. It's probably going to take us quite a long time to run through. But this is very interesting. We do this in the hospital every single day. When someone's sick, we take a swab, we collect body, either fluid samples, tissue samples. We look at it under a microscope to look for some of these organisms. All right. Let's jump through it. So what do we need to know for this dot point, Matthew? What are the key things? So just dumb it down for us. If I was your friend at school and I asked you, what do I need to know? What would you tell me?
Is this for 1.1? Because
Yeah.
I'm still stuck on trying to find it. Just this on the screen?
Yeah, just on the screen.
Oh, I don't know.
It's hard. It's a lot, right? So let me
Yeah,
help you
talk
all,
about-
right? First thing you need to know is what makes all of these guys different, right? How do they look different? What do they do different? Okay? What makes something a bacteria? What makes something a human? What makes something a plant? The next thing you need to know is a very brief overview about these processes. So how do they reproduce? So these are the listed methods of reproduction for every one of those organisms. Now, one of the most important things you need to be able to do for this dot point is to define sexual versus asexual reproduction, right? What does it mean? What's the difference? And you need to tell me which one of these are asexual, which one of these are sexual. Okay? Very important. That's how the syllabus is going to test you. That's what your exam questions will all be about. So the typical exam questions you'll get is they'll give you a picture of a bug. They'll say, here is an example of a unicellular organism with a cell wall, one loop of DNA, and this funny looking tail. Can you tell us how it replicates and identify whether it's sexual or asexual? That is your textbook exam question. Okay? So let's start with the first part, which is what makes these organisms different. And for that, you need to tell me what the word prokaryotic and eukaryotic means from Year 11, Module 1. So let's go to Atul. Atul, what's the difference between prokaryotic versus eukaryotic?
It's the presence of membrane-bound nucleus, a membrane-bound nucleus, and membrane-bound organelles.
Good. Why do we make that difference? Which one's older? If I showed you a cell,
Prokaryotic.
very good, right? I remember prokaryotic means primitive. And the reason is to form membranes took many, many millions of years of evolution, right? A membrane is a barrier that protects an internal compartment from an external compartment. That's quite complex. So it took millions of years in evolution to develop. So remember, prokaryotic are primitive or old cells. So they lack a nucleus, and no membrane-bound organelles. Now, this is where students trip up. Those of you who rote learned, which is fine, I'm gonna take the rote learning out and show you and understand how to make sense of bio, will struggle to define what membrane-bound organelle means. And what's a membrane-bound organelle? What does that mean, and what are some examples?
I don't know how to explain it, but I know ribosomes aren't one, even though they do have like a membrane. It's like they have their own like fluid. So
Okay. Good point. You're completely right there. So I really like how you picked up a very high point, which is the only organelle that is not membrane bound that you need to know is the ribosome. Everything else is bound by a membrane. The point I would say is we need to reconsider is that ribosomes don't have a membrane. They don't have a membrane at all. If you look at a ribosome zoomed in, I know all of you see these little balls, right? But that's a model. That's a visual representation of something. That's not what a ribosome looks like. Ribosome is literally like a piece of string, right? It's made out of RNA, which is a variant of DNA, right? So the ribosome literally just looks like this. It's a bunch of, if any of you have wired earphones, it looks like it's been tangled in your pocket. That's what a ribosome looks like. It has 2 units and it looks like this. Of course, you can all appreciate this is not bound by anything. There's no compartment. There's no case to this. Other organelles have a case, right? So like an AirPods case, you have organelles. I'm going to draw this one. It looks like a Wi-Fi signal. What organelle am I drawing right now? Someone shout it out. What is this organelle?
Golgi.
Very good. The Golgi apparatus. It's the Wi-Fi signal. It's the transporter of the cell. It's the postal package. It sends vesicles that will butt off, go inside, outside a cell, different parts of the cell. It is bound by a membrane, because if I zoom in, I can clearly see there is a barrier between the external and internal environment. That's what membrane bound means. So I want you all to appreciate membrane bound just means the organelle is compartmentalized. It's closed off by something, right? The ribosome is not. It's just a bunch of strings that are connected to each other. Very good. Now, which one of these organisms is prokaryotic? Let me go to, let's go to Sarah. Which one of these organisms is prokaryotic? There's only one. It's your little hint. Any idea?
Bacteria.
Very good. One of the oldest life forms. Right on your pen tip, you all have billions of bacteria that are looking right back at you. We just can't see them, right? So very primitive old organisms. They don't have a nucleus. They don't have any membrane-bound organelles. Very good. Every other life form that you see here, protists, fungi, plants, and animals, are all eukaryotic. Okay. Now, the way I'm going to structure this teaching is I'm going to tell you exactly what a bacteria looks like. I'll show you how it reproduces, and then we'll talk about asexual versus sexual. And then we'll go to the next life form, the next life form, and the next life form. So that's how I want you to head out your notes. Okay? Before I tell you more about bacteria, we need to answer this question. Many students have the biggest misconception about what it means to have a sexual reproduction versus an asexual form of reproduction. So, Nathania, do you want to just give it a go? What's your understanding of something being defined as asexual reproduction versus sexual? Any ideas, Nathania?
I think sexual reproduction is a combining of genetic information.
Good. You're very good, right? It is a combination of genetic information. But you see, I completely understand you haven't learned this yet, right? So really good job with that answer. It's just the genetic information part is a bit vague for us. So I'll help you all define this further. But does anyone else want to posit sexual versus asexual? What does it mean? Or what are some differences? I just want to know a baseline understanding. Atul?
In sexual, doesn't gamete formation occur?
Very good.
So
Very
then
good.
the variation?
Good. Atul, have you learned this already or?
No, not
No.
yet.
Okay. Very good. Good job, right? So he went right through the misconceptions. Usually students tell me, over the years I've seen many answers to this question. They usually tell me sexual involves 2 parents, asexual involves 1, right? And that's not true. You can have sexual reproduction. An organism can sexually reproduce with itself, and the example is in plants. Plants have both male and female reproductive parts. So they can make the male and female reproductive gametes, combine them, an offspring, right? So it's not about the number of parents. I want you to understand that. Do not ever talk about number of parents. There's a general rule of thumb that sexual usually involves 2, asexual usually involves 1, but that is not a law or a defining feature. Now, the defining feature is genetic variation. Sexual reproduction involves genetic variation. Asexual reproduction involves no genetic variation. If you forget everything today about bacteria, how they look, what they do, this is the one thing you can't forget. Because this is your principles. If you forget this, you will not be able to build upon this knowledge. I'm going to ask you all this many times. Sexual, genetic variation. Asexual, no genetic variation. So now here's a follow-up question. You all just briefly learnt about the one cellular process that allows genetic variation to occur. What is the name of that process? Atul alluded to it. He said we have the formation of gametes. What is the name of the cellular process that gives rise to genetic variation?
Meiosis.
Very good. Good job. Good. You're all doing really good with the answers. I'm very happy with how much everyone's contributing. So very good. Keep it up. Now, perfect. Sexual involves genetic variation, and sexual reproduction always uses meiosis, which means we produce gametes. These genetically varied organisms are sex cells that have half the genetic information. Good. All right, I think we're ready to move on. Now let's talk about bacteria. This little critter here, right? Now, just a bit of an aside about bacteria. Can someone tell me a nasty disease caused by bacteria? In hospital, there is a very common disease caused by bacteria, which we'll talk about, but off the top of our heads, can anyone think of any nasty diseases or any diseases at all that bacteria cause? Say it off the top of your head. Let's go with Sarah. Give me a disease caused by bacteria that you might know of. Any diseases at all?
I'm not too sure.
That's all right. Ed, give me a disease caused by bacteria.
Oh, I don't know one.
That's all right. All right, let me give you a one. Has anyone heard of pneumonia? Pneumonia. Anyone heard of that? Does anyone know what that is?
Yes.
Okay, now if any of you go into medicine, this is probably going to be the most common infectious disease you will come across on top of colds, right? Pneumonia is infection of the actual lungs. So it's not an airway infection like with colds and flus. Pneumonia, the actual air sacs, the alveoli get infected. And it's by these bugs. It is so common. And in developing countries, people die from it. And little kids and old people die from pneumonia as well. In fact, the reason we were so scared of COVID, everyone, is not because of COVID directly, but it's because COVID destroyed the airways and it allowed bacteria that existed in those airways to divide, replicate, destroy the lungs, and eventually stop someone breathing properly, which resulted in death, right? So bacteria cause a lot of really fatal, nasty diseases. There's so many more that I can talk about, but we'll come across that when we talk about Module 7, infectious disease. Good. This is what a bacteria look like. Now, hopefully you can all appreciate it's prokaryotic. And how can you tell, Alyssa? When you look at this cell, what features are telling you this is prokaryotic or not eukaryotic?
There aren't any membrane-bound organelles.
Very good. And remember, as Anne mentioned, the only membrane-bound organelle is a ribosome. This is a huge exam-relevant point, guys. If they show you a cell and only has ribosomes, you can probably bet it's prokaryotic, right? The other thing you notice is the DNA is not enclosed in the nucleus. In fact, even though it looks like spaghetti soup, right? In fact, if you unwound everything, it's just one loop of DNA. And that one loop is called one chromosome, right? We spoke about chromosomes last week, and I'll give everyone the recording to what chromosomes are. But essentially, if you get DNA and you wind it very tightly into a long structure, it forms a chromosome. So chromosomes are made out of DNA. Okay, good. Now what you also see is these little pili. That's for adhering to cells, right? So these little pili are like little arms that bugs use to catch onto tissues in your body or invade through. And this flagella is like a tail. It's a motor. Hopefully you all know you're all 60% water. So this flagella is like a motor that allows the bacteria to swim through your body and cause infection in different parts. Very good. So now you all understand the structure of bacteria. Can someone tell me what is the unique feature that I have not spoken about yet? Because this is also one of the key defining features of bacteria. What do you all see? Looking at it under a microscope. And Ed, what's the unique feature I haven't spoken about with bacteria?
Not sure. I mean, different types of cell walls. That's one.
You said it, cell wall, right? Cell wall. There's only one key type of cell wall for a bacteria, but cell wall is a unique other feature we haven't spoken about, right? The pili or the fimbriae, the flagella we've spoken about, it's the cell wall. So if you see a one-celled organism that's prokaryotic, so these are the defining features of a bacteria. All bacteria are unicellular, aka they're only one cell. All of them are prokaryotic, and all of them have a cell wall. A cell wall is kind of like a protection. It's like armor, right? This is why some bacteria don't work on some antibiotics, which are medicines we use to kill bacteria specifically, don't work on some bacteria because they have a very thick coat of armor that prevents any drugs getting into their body, right? Now, the cell wall is made out of a compound called peptidoglycan. You do not need to know the name of this, right? So I shouldn't even be writing it in red. It's very low yield. You don't need to know that this specific name, peptidoglycan. The reason I'm telling you is it tells you what it's made out of. Matthew, where have you seen these words before in life? Peptide.
In proteins? Isn't
Very
that part
good.
of proteins?
Very good.
What's
So
that
now you know
part?
cell wall is made out of protein. And what's the other word? Glycan. What does that remind you of? Anything?
Sugar.
Very good. Glycan is, look at you guys, teaching yourself just off these names. Very good, right? Now this you need to know. The bacterial cell wall is not made out of cellulose like plants. It's made out of protein and glucose. It's the protein that's the most important. Okay? As long as you can tell me that, very good with bacteria. Very good. All right. I think we can move on. Does anyone have any questions about what a bacteria looks like? Okay. Now let's talk about how a bacteria divides. It's quite straightforward, actually. I'll show you the formal image because it's lower down in my notes. Yeah. This is a mode of replication of a bacterium, right? Now, the name is called binary fission, and it's going to tell you a lot about how this bacteria replicates. Now, in an exam, the key things you need to say is that the bacteria will elongate, okay? It'll form this barrier separating the two cells, known as a cross wall. The other thing you notice is that the DNA or the genetic information doubles. Genetic information, DNA, doubles. DNA doubles. Okay? And there you make two daughter cells that split off into two. Now, what does binary mean? For those of you who like computer science, or can you tell me what binary means? Atul, what does binary mean? It's the word binary. Two. And what does fission mean?
Split.
Very good. So the word binary fission literally means to split. And binary are numbers, right, that have equal magnitude or importance. So that's telling you that binary fission is a very symmetrical process of splitting a cell into two. This is technically mitosis. The reason we don't say this is mitosis, and you should never ever say that, is because mitosis is for eukaryotic cells, right? Mitosis is for cells where the nucleus has to disappear. Spindle fibers need to form. The chromosomes, there are many chromosomes that line up in the middle of a cell. You might have seen that before. None of that's happening here. This is a very simplistic doubling. It's more primitive or basic than mitosis. So it's not mitosis, okay? But it has a lot of commonalities. Good. Now, I want us to think logically, right? You have just seen how a bacteria divides. If you were to create an antibiotic, so you're going to start your own pharmaceutical company, create a new antibiotic to save lives, what are some mechanisms you can target and stop a bacteria doing to kill it? Very logically, looking at this. Let's go to Anne. What is something you can stop or block that would prevent a bacteria dividing?
You could probably stop the DNA from replicating or stop cross walls from forming.
Perfect, right? And that's literally how antibiotics work. They accumulate in the cell wall of a bacteria and they kill off the bacteria. The other way antibiotics can work is you can go to the DNA of a bacteria and stop it replicating. And then when you look at it macroscopically, you look at the person taking an antibiotic, they get better and their life is saved from a serious bacterial infection. Now, here is an extra question that will show me who's thinking and who's, I want everyone to be thinking, so all of you will get the answer to this. Which one would be more harmful to humans? An antibiotic that stops DNA dividing, or an antibiotic that blocks the cell wall dividing? Any ideas? And this comes from your understanding of what a human cell has versus a bacteria cell. So think about, do humans have DNA? Do humans have a protein?
Wouldn't it be the wall? I mean, the one that's more harmful would be the DNA, the wall.
DNA, right? Why do you say that at all?
Because animal cells don't have a wall.
Very good. So if you create an antibiotic that destroys a cell wall, it's not going to affect us because we don't have a cell wall. So most antibiotics that you've all taken are cell wall destroying antibiotics. The antibiotics that stop DNA dividing, they have a few side effects for humans, right? I'll give you a key example of this. If you give a woman this specific antibiotic called trimethoprim, it stops DNA dividing. If the woman is pregnant, that can end the pregnancy. That is how severe the side effect of that antibiotic is, because remember, a baby inside the uterus is rapidly dividing, creating a lot of new DNA, right? So hopefully you're understanding everything you're learning here. We apply as physicians, as surgeons, as doctors, when we're prescribing medicines to patients as well, right? Very good. All right, let's move on. Now let's talk about protists. Now, there is one famous protist that has killed about one child every 5 minutes from starting today's lesson. It is a protist that lives inside the digestive system of mosquitoes, and that everyone should take medications for before traveling to countries that have this specific disease. What am I talking about? What disease?
Malaria.
Very good, right? Malaria is caused by a protist. And we're going to talk about that later in Mod 7. Now, my question now is, what is a protist? How is it different to a bacteria? They look similar under a microscope, but they have some key differences. Let's go to Alyssa, since you've mentioned malaria. Any ideas what makes a protist different to a bacteria?
It's not a prokaryote. It's a eukaryote, but it's unicellular.
Very good. This is literally the key thing. The reason why everyone gets confused about protists is they're kind of like a mismatched family. They consist of a wide variety of completely different living organisms. The only uniting feature among them is all of them are one cell in size and all of them are eukaryotic. Okay, apart from that, they're different in every way, shape, and form. A lot of protists don't cause disease. The ones that cause disease have a variant name called protozoa, which is what we call the bug that causes malaria. It's a protozoa. It's a protist that causes disease. That's what protozoa means, right? So unicellular eukaryotic. Now, the other key defining feature is that a protist, right, unlike a bacteria, does not have a cell wall. So if you ever see a single-celled organism that has a cell wall, it's probably also prokaryotic and it's a bacteria. Okay, so unicellular eukaryotic does not have a cell wall. This is an example of a protist that you see, right? So it's all one-celled. It's just the one cells are dividing. These are all copies of that one cell in all different shapes and sizes, which we'll talk about shortly as well. Very good. Now, I'm going to show you a way to remember all the methods of reproduction, because have a look at this dot point, right? If you go all the way up to page 20, is it 22? You will see dot point is hefty. Where is it? All the way up here. You need to memorize all of this. Asexual, resexual, budding spores, binary fission, budding, which organism does what. The way you remember this is in my head, I move through increasing order of complexity. So the simplest one is bacteria, and it can only do binary fission. Now, the second most complex organism on this list are protists. Now, since they're slightly more complex, the way I remember this is protists can do everything that a bacteria can do, plus they can do one new form of reproduction, kind of like a leveled up bacteria. It's in my head what they're like, right? So if we go back to protist, we'll talk about budding, which is the new cell division process. Now, brief question, this is something I should have highlighted more to you all, which is this image here, binary fission. Bethany, is it sexual or asexual and why? Binary fission. Is it a sexual form of reproduction or an asexual form of reproduction and why?
Asexual.
Okay. Good, Ed. Rithania, why is it asexual?
Because there's no genetic variation.
Very good. Right? DNA is replicated. One becomes two, but it's identical. Then it splits into the daughter cells, the bacteria, right? And it's exactly identical. There's no genetic variation. There's no meiosis happening here. And so this has to be asexual. So all of you next to binary fission, write asexual. Now let's talk about budding. Budding is very simple as well, right? Budding is literally this very similar to binary fission. The only difference is you start off with a cell, and compared to binary fission where a cell would just elongate, form a cross wall, and split into two evenly sized cells, in budding you form a bud, so a little outgrowth, right? This is an outgrowth of the cell membrane. Do you see how it's asymmetrical? It's like forming a tumor on your head. That's literally what a protist does. It forms a little outgrowth or a vesicle. And then it doubles everything. So it doubles its DNA. So whatever DNA it had, it will double it and throw it inside this vesicle. Whatever organelles it had, the Golgi apparatus, the endoplasmic reticulum, its ribosomes, it'll also throw in a couple of everything inside. And eventually, that bud will then close off and it will split. Now, let me ask you, is this a symmetrical form of reproduction or division like binary fission, or is it asymmetric? Let's go with Ed. Symmetric or asymmetric?
Asymmetric.
Yeah, very good. So it's asymmetrical, right? Good. That's the key difference. Now, Anne, I see you have a question. Let me ask you a question first. Is this sexual or asexual?
Asexual.
Very good. Because you just copy DNA, right? It's going to be the exact same. Good. Now, you had a question?
Yeah, so you know how you said protists also have binary fusion?
Yep.
In the picture above, it shows the cross walls forming, but because it's a protist and it doesn't have cell walls, is it the same?
Very good question. They would not. So the cross wall is just a partition between the cells. So for a bacteria, the cross wall is going to include the cell wall. For a protist, yeah, you're right. You wouldn't technically call it a cross wall. So for a protist, you'd say binary fission would just involve symmetrical elongation and splitting of the cells. I wouldn't mention a cross wall. Very good pickup.
All right, thank you.
Yeah, very good. Good. It's always good when, even after teaching for so many years, when you guys ask new questions that students haven't asked before, because it shows that you're all thinking very critically. So good. Good. All right. So protists, we've spoken about budding. We've mentioned it's also asexual, but the key difference is it's asymmetric as well. Very good. Now, before we continue off and we start talking about fungi, which is going to be our next organism, we've hit the hour mark. So what I get you all to do is go stand up, stretch, walk around, get some water, take a 5-minute break. The reason being, if you sit for 2 hours non-stop, you send rest and digest signals to your brain, and that's why you feel sleepy, especially in long classes. So go take a break. When you walk around, blood flow to the brain increases too, so you get more attentive. Now, sorry, before you take your break, a bit of a small point about those of you who want physical copies of these notes as well. Can you all hit the raise hand icon if you want a physical copy of these notes? Okay, good. All right, so most of us. So for this, the only issue is we can't send it online for you guys to print, etc., only because of copyright issues and distribution, etc. So the plan is going to be, I will be messaging probably your parents via email about getting access to this physical copy of the notes. Now, once I get confirmation from all you guys, and hopefully you can do it ASAP, I'll send it by tonight. So just remind your parents to check their emails ASAP. The next day, what I'll do is I'll go print out the notes. They're about 200 pages for a module, right? So it's going to be for every student, it's going to be thousands of pages. So it'll take some time. I'll print it all out. I'll bind it for you guys. And then by the end of the week, you can come pick it up, possibly directly off me as well. So I will either be in the city during most of the week after hours, or I will be, I work at hospital near South Sydney, near Cogra. So you guys we can talk about where you can pick it up. But ideally, when you have time, you can, if you're near the city, you can quickly pick it up from me. That's going to be the plan. Okay. Any questions about the physical copy of the notes? All right. Good. Okay. Well, take a 5-minute break. We'll recap at 3:10. Okay. Good. See you guys then. Okay, when we're all back, just hit the raise hand icon so I know, and we will continue. Good. All right. Let me pull up fungi. Does anyone have any questions about Year 12 exams, your careers, futures? Feel free to stay back after class if you want to individually discuss that as well. I'm more than happy. Yes, let's go down to this. All right, so we've spoken about bacteria, we've spoken about protists, and the next point leads us to fungi. Now, fungi don't normally cause human diseases because they don't have too many advanced ways of affecting the body unless the body's immune system is disrupted. That is when you'll start to see fungal diseases happen in humans. Now, does anyone know any examples of any diseases that weaken the immune system, or any fungal diseases for that matter? There's one infectious disease which directly attacks the human immune system. There is no other infectious disease that does that. It's like a robber that goes and attacks and robs a police station. That's how bold this disease is. It's 3 letters, the name of this disease. Does anyone know what I'm talking about? There was an epidemic in Australia in the 1980s. I'm talking about HIV, right? So HIV weakens the immune system and increases the likelihood that fungal infections will occur. So that's when, as a doctor, we start thinking about fungi as a cause of disease. Good. Now, let's talk a bit about fungi. Fungi are a bit more complex, so the next level up. Remember what I told you all? Every level up in terms of complexity of life, we take one form of reproduction from the previous level and we add on a new form of reproduction. So with bacteria, Rithania, what was a form of reproduction for bacteria?
Asexual?
Yeah, but what was the name? True, it is asexual, but what's the name of the reproduction process?
Oh, binary fission.
Good. And Alyssa, what do protists do? So they do 2 forms of reproduction. So you take the previous one, which is binary fission, and what are you going to add on?
Budding.
Very good. And then finally, we're left with, let's all say asexual, fungi. What fungi are going to do is they're going to take the complex previous reproductive process, and then they will add a new one. Now, has anyone stepped on a mushroom? Very weird, random question, but it does relate to this point. Has anyone stepped on a mushroom on grass, and did you notice anything unique happen afterwards? Anyone who's been curious with grass and nature? And anyone seen a cloud of smoke come out when you step on a plant or a mushroom or any of that sort? Hit the raise hand icon if you have. You don't need to verbally tell me. No one? Okay, well, good. Thanks, Ed. Makes 2 of us. So mushrooms are prime examples of fungi. What I was trying to do is relate life to biology, because that's literally what biology is a study of. So if you've lived, 15, 16 years, you've actually done a lot of biological research just by being curious, right? So mushrooms, they release what's called spores, which are little particles of new life form that will then disperse in the air, travel really far, plant themselves in the soil, and form a new fungus, which in this case would be another mushroom. Okay? So given that mushroom are an example of fungi, let me ask you this. Are fungi unicellular, multicellular, or both? Matthew, what do you think? Fungi, unicellular, multicellular, or both?
Maybe multicellular?
Okay, so they definitely are multicellular. That's one thing, because mushrooms are microscopic. But another very common example of fungi is the living organism which we use to make alcohol? Does anyone know what the name of this living organism is? Those of you doing chemistry, you will eventually must learn about this.
Yeast.
Very good. Atul, what makes yeast unique? Are they unicellular or multicellular? Can you see yeast? It's a simple question.
No.
No. So they're unicellular. So that's telling you that fungi can be either unicellular or they can be multicellular. Okay? So next to fungi, I want you to highlight a very important point. It can be unicellular or multicellular. They're all eukaryotic. Remember I told you everything other than bacteria are eukaryotic. Okay? What's the key difference between a fungi and a plant, however? If I showed you an example of a unicellular fungus, it's eukaryotic, so it has a nucleus, right? It has other membrane-bound organelles. What would make a fungi, a unicellular fungi, different to a protist, which is also unicellular and eukaryotic? Ed? What makes a fungi different to these other life forms? Protists specifically.
Did you say Ed or Anne?
Sorry? What was that?
Did you say Ed or Anne?
Ed. I'll say Edward.
But heterotrophic, maybe.
Good, but protists are also heterotrophic. They obtain nutrients from their environment. That's what heterotrophic means, everyone. Autotrophic is you obtain energy from the sun. Heterotrophic is from the environment. How is a fungi different to a protist? Any clues? Anyone? Okay. The key difference is this.
So would it have a cell wall or?
Very good. It has a cell wall. Good, right? That is a single different feature between a fungi and a protist. That's how they'll trip you up. They'll show you an example of a single-celled eukaryotic organism, and you'll be trumped on whether it's a protist or whether it's a fungi. And the way you ask or answer that question is ask yourself, does it have a cell wall? I clearly remember this being an exam question when I sat the HSC. So it's high yield to know this point. Okay, so fungi have a cell wall. The cell wall is made out of a protein called chitin. C-H-I-T-I-N. Chitin. If you want to be different, you can call it chitin. Doesn't matter. But let me show you an image that's a multicellular one. Let me just draw it out for you. Okay. So here, single-celled nucleus, other membrane-bound organelles like the endoplasmic reticulum, which I'm drawing here. What does the endoplasmic reticulum do, Sarah?
I'm not too sure.
It's like a conveyor belt, right? So it's a factory. This is just a surface, this weird squiggly lines that you're seeing, kind of like moving conveyor belts. And the factory workers are your ribosomes. They sit on that conveyor belt and they start doing their work. And Alyssa, what do ribosomes do?
Synthesize protein.
Very good. So we say that an endoplasmic reticulum that is a rough, aka it's peppered with ribosomes. So if you were to touch it, you'd feel like you're touching something that's a very rough surface, right? So say it makes protein. And when it doesn't have ribosomes, it makes fat and it detoxifies or breaks down drugs. Very good. All right. So coming back to that, that's what a fungi looks like. The key feature is it has a cell wall made out of chitin. Good. Now, let's talk a bit about spore formation. Now, maybe I'll draw it myself because none of these diagrams from the textbooks are my favorite. Okay. Now, when a fungi engages in spore formation, it can do two different kinds of spore formation. I want you to pay attention to these two scenarios. I'm going to ask two of you at random to tell me, is it sexual or asexual, right? So we have one form of spore formation. In this form, what happens is you will have a fungus. I'm just drawing it like a little long stalk. It can look however it wants to. It can look like a mushroom for that matter. It will make a spore, and the spore is going to be diploid. Now, we've said the word haploid, and we said haploid means it has half the genetic information that it normally should have, right? So a human has 46 chromosomes. So, Atul, what would the haploid number of chromosomes be for a human cell?
23.
Exactly, right? So this is a diploid spore, meaning it has a full set of chromosomes, right? It's not 46, because this is not a human that we're looking at, it's a fungus, right? Weirdly, simple organisms seem to have more chromosomes. It's really weird. Tomatoes, for example, have more chromosomes than us, which is weird, because they're just a tomato, right? Doesn't really make sense. We haven't really answered that question yet in science. But this is a fungus. It has, let's say 8 is the diploid number, right? That's a full set of chromosomes. A diploid spore would also have 8 chromosomes. So what would happen is air, wind, for example, would disperse this diploid spore. And after some period of time, it will grow into another fungus. Now, is this sexual or is this asexual? Let's go to Rithanya.
It's asexual.
Good, very good, right? And it's because when you make a diploid spore, it was made from some variation or form of mitosis. So you genetically just copy the information, so it'll be identical. Very good. The other example, this is an asexual form of spore formation. The other formation, for example, is when you make a haploid spore. So just a little point, guys. We write 2n to show the diploid number of chromosomes, and we write n, you'll see this in textbooks, to represent a haploid or half number of chromosomes, right? The other form of reproduction would be when you have 2 different fungi that each make a haploid spore, right? And each of the haploid spores will combine to make a new fungus. Now, is this sexual or is this asexual? Let's go to Sarah. Sexual or asexual?
Sexual,
Why do you say that?
because 2 of them are combining.
Good. So is there genetic variation or no genetic variation?
There's no genetic variation.
Think about this.
No, there is genetic variation.
Very
Yes.
good, right? Because fungus 1, Mr. Dad fungus, and then fungus 2, the mother fungus, are 2 different plants, right? I shouldn't say the word plant. They're not plants, they're fungi. But they're 2 different organisms, so they have a different genetic makeup. So when they each contribute half, the resulting offspring is going to be different. So the key thing is spore formation can be sexual or it can be asexual. Okay, very good. Good. Now, another term, just one term I'd like you all to know, is this word called hyphae. Forget about this whole rhizoids, stolon. If you memorize this, you're just wasting brain space. More interesting things you could be learning, right? Hyphae is the important thing. A hyphae is just a reproductive tentacle of a plant. So a hyphae contains spores that are going to seed off and form new fungi. It could be haploid spores, it could be diploid spores. Okay? Good. So just know hyphae is a reproductive structure of a fungus. Reason this is important is when we look under a microscope, if we see these hyphae, we go, okay, this patient has a fungal infection. It's one of the key defining features, reproductive structure or branch of a fungus. Okay, good. Let's go to, you can have a read of these in your own time from the notes, just a little bit more like terms and cycles. Do you need to know this? No. Okay, can they show you this diagram and ask you to explain it? Yes. Okay, so I'd say there's no harm in reading this in your own time, but don't commit it to memory. Just understand what's going on. So if you are shown a diagram, you can just talk about it briefly. And question?
Yeah, I just wanted to ask, for diploid cells, are they double the number of chromosomes of a normal cell, or are they just...
They're
Okay.
just normal, yeah. So it's weird. Diploid has a D, it could mean double, I get where you're coming from. Yeah, diploid is just a normal number of chromosomes. So haploid is half that. So for a human, diploid number of chromosomes is 46,
Yeah.
haploid is 23.
Okay, so diploid is when it's got the full set of chromosomes.
Right.
But it's double of a haploid.
Exactly. Double.
Okay. All right,
Right.
thank you.
No worries. Good clarification. Does anyone have any other questions? Feel free to ask. I love questions because it shows that you're all thinking critically. Good. I remember when I was in Year 12, I had so many questions. It got to a point where my school teachers did not know the answer to the questions. I remember one of my teachers telling me that bacteria were eukaryotic because they saw the word eubacteria in a textbook. So please ask anything and everything, clarify it. It's a rare opportunity because you won't be able to ask many other people very specific niche biology questions. Okay, good. Let's go to, gone through bacteria, we've gone through protozoa, we've gone through fungi. Let's talk about plants briefly. What does a plant look like? I'm not going to show you anything. I want you to visualize it and tell me, what is a plant and what does it look like? Let's go to Matthew. Matthew, what are some key defining features of a plant?
The carpel, the stamen, the petal, the sepal.
Okay, very good. So we're talking about the reproductive structures. Very good. Let me ask you a follow-up question. How about the cell of a plant? If I pulled a plant cell out, it's an image there, but now, what are some key things I'd see under a microscope for a plant cell?
The chloroplast and chlorophyll. That makes a
Very
difference.
good. Very good. So that's what makes a plant green. Ruthania, what's another key feature I'd see in a plant cell? So we said chloroplast, chlorophyll. Very good.
Stomata.
So stomata are seen on the underside of a leaf, right? So they're a structure. I wouldn't call them a cell. When you look at a cell, a stomata is literally a hole, and there's two guard cells on each side of that hole, and they control the opening and closing for gas exchange, right? So it's kind of like an entrance to the, entire plant itself. What about a plant cell? That's all. I'm looking for one word, and I know you all know it. I just want you to say it, which is a hard part about bio. There's so much stuff. You need to tell me the most important things in an exam. So, Atul, what is the key feature of a plant? Cell.
I'm not sure.
So we said it's green, the chlorophyll, chloroplast. What else does it have? Looking for one more thing.
Mitochondria.
Vacuole.
Very good point about vacuole. So animals also have a mitochondria.
Is it the cell wall?
Yes, very good. That's what I'm looking for, right? All of you did very good, right? All of you answered very good points. Vacuoles in plants are humongous because plants can't go to the kitchen and get water like I did on my break, right? So they need to have huge vacuoles to just store water, especially if they live in Sydney or Australia where it's always hot most of the time, right? Except for the recent weather. So plants have huge vacuoles. They have chloroplast, chlorophyll, and the last thing I was looking for was a cell wall. If you tell me those three things, you understand what makes a plant up. And as you all know, it's multicellular and eukaryotic. Okay, so this is what I'm trying to show you. As you can see, the huge blue puddle, that is a vacuole. It is much smaller in animals because we don't need to store that much water. The key thing is it has a cell wall. What is it made out of? Not protein.
Cellulose?
Very good. And then here's a follow-up question, especially for those of you who do chemistry. What is cellulose made out of? It's very interesting because right now, one of the key dilemmas in science is what are we going to do when petrol and fossil fuel runs out, right? Elon Musk has partly solved that problem with electric vehicles. But right now, that's still using burning fossil fuels, but we can eventually switch to solar fuels. But a new exciting avenue is cellulose, right? If someone discovers how to break cellulose in its bonds and use the resulting molecule to make energy, they will change the world. Just we haven't figured it out yet. Who can tell me what makes up cellulose? What is the molecule I'm talking about that when you burn it, you'll make energy? I'll give you a hint. You need 50 grams of this a day. If you have more, you'll get diabetes.
Glucose.
Very good.
Oh,
So,
sorry.
yeah, no, good answer. It's glucose, right? So cellulose is a long chain of glucose. Good, that's it, right? So it's just sugars that are connected together to make a long chain. So now you know the structure of a plant. Edward, can you just repeat those 3 key things for the rest of the class?
What was that? Sorry?
What are the 3 key components of a plant cell?
Cell wall, chloroplasts, and the vacuole.
Good, right? Large vacuoles. Now, I'd say in order of importance, these are the 2 things I care about. If you say the 3rd thing, it looks good. That's not going to be in a marking guideline, okay? Let's talk about how a plant reproduces. Now, this is what Matthew was talking about when he explained the key features of a plant. Now, plants are very interesting because I thought, you know what, plants are pretty stupid because they make fruits and then they just give away their nutrients for free, right? Doesn't make any sense. But you'll realize later that the creation of fruits and plants is using animals to disperse seeds, right? Animals will eat the fruit. Inside the fruit are seeds, and then the seeds will travel kilometers away as a plant walks away. And then when it's released into the environment, it will form a new plant at a far distance away from the parent, so there's no competition. So they're very smart that these essentially non-conscious things, organisms, are using animals to proliferate and continue their species. Now, the other interesting point is that the flower is the sexual or reproductive organ of a plant. The flower is equivalent to the gonads, the testes and the ovaries of a male and female, right? So let's have a look at these names here so we understand it. Now, these little projections that you see that are golden there, those are the male reproductive part of the plant. And that long central green tube is the female reproductive component of a plant. You can straight away realize, compared to a human, they have both male and female reproductive components. It's a little weird, right? So all plants are technically intersex. It's the difference versus humans. Does anyone know what percentage of humans are intersex? AKA they're born with both male and female reproductive parts. Any guesses? It's 1%, which means if you know 100 people, at least one person, statistically speaking, was born with some degree of male and female reproductive parts. So it's actually a very common phenomena and a genetic disorder that is treated later on in life. Coming back to this, the keywords I want you to remember is the stamen for the male part of a plant, stamen, then the carpel for the female part of a plant. Now, you don't need to use all of these terms. In my exams, all I've ever used is anther, and I talk about the stigma, style, and ovary. So pick some key words to remember. I would say the stamen, and I would say the carpel are the most important. Okay? Have little mnemonics, ways to remember this. Maybe you can remember stamen has men for male, right? And then carpel must be female by exclusion. I just made that up. But pick some mnemonic or way to remember these fancy words. Otherwise, you're going to forget it all. Okay, so looking at this part, we've got the anther at the tip. So this is part of the stamen. And at the tip of this anther, it's coated in pollen. Now, Matthew, what's the human equivalent of pollen?
Sperm.
Very good. So pollen is the male sex cell, right? So it's a spermatozoa. And what happens is a bee or a pollinator, it's usually a bee or some other insect, will come across, take the nectar in that plant in the center, and whilst doing that, it's going to brush its body against all this pollen. And then when it moves to the female part of the plant, it's going to drop some pollen grains down this tube, right, which is called, so this tip is called a stigma. The tube is a style, right? And it kind of falls down this tube, and it goes to the ovary of a plant, right? And what's in the ovary? It's the female reproductive cells. Inside are the ova, which are the female reproductive cells. Interestingly, the ova is the exact same word for human, female, like the female sex cell from humans. They're also called ova. It's just the pollen is named differently to spermatozoa. Okay, so once it travels down, it's going to fertilize the ova. Now think about simple maths here. The ova has half the genetic information. The pollen has half the genetic information. When they combine, do we end up with a diploid or do we end up with a haploid cell when the pollen and the ova combine?
Diploid.
Good. And Alyssa, is this sexual or is this asexual reproduction, and why?
It's sexual reproduction because it involves the gametes fusing together.
Very good. And how were the gametes made, Alyssa? What process?
Meiosis.
Very good. So every single pollen of this same plant is going to be different to the next pollen. So if I take 100 pollen from this one flower, every single pollen grain will be genetically different. If I take 100 ova from this same flower, all 100 will be different. So when you combine each pollen and combine each ova, of course you end up with millions of combinations, right? If you do the maths, if your parents were to have children, they could have, to even more children speaking, they would have more than, I think it's 20 million combinations of gametes or offspring that you can have, right? So there's so much variation that can arise from this. And we'll talk about how that variation happens later. For now, I want you to just accept meiosis has variation, and later I will teach you how that happens. But very good. Now, this is called self-pollination, aka self-pollination, aka the gametes both arise from the same plant. What can also happen, everyone, is a pollen can go and travel to another plant, completely different plant, travel down the stigma, style, and ovary. What would you call that? Has anyone heard of the term for when you pollinate from one plant to another plant?
Cross-pollination.
Very good. Cross-pollination. Both are sexual. Pollination is sexual. I want you to remember that. The biggest mistake students make, and examiners know this, so they will test you on this, is they'll say self-pollination is asexual because it involves one parent. That is a huge misconception. And hopefully all of you can remember that and maybe write it down in red asterisk. Pollination equals variation as gametes were used, therefore sexual. Something along those lines. Perfect answer to that question. Good. Any questions at all? Now, let me ask you, what happens after this, right? So what do we call the process when two gametes meet? What is the name of that process? Two gametes meeting. It happens in the human body too. It happens in the outer 1/3 of the fallopian tube. Let's go to Rathania. Rathania, what is the name of the process when two gametes unite?
Reproduction.
Fertilization. Fertilization is a name when both gametes combine to form a new cell. And what is the name of that cell that is made? So what I'm explaining now, everyone, is the two gametes combining that both have n number of chromosomes to form a new cell that has a 2n number of chromosomes. That is called fertilization. Fertilization is the union of gametes. Second question is, what is the name of this one cell?
Zygote.
Very good. Zygote. Zygote is the one cell that's made from fertilization. So that's just a further terminology. And in layperson terms, the zygote is the seed. So now all you need is for the seed to get away from the parent. So guess what the plant does? It coats the seed in sugars, makes it colorful to attract animals, which will eat the seed, and historically will then excrete it in the land far away, and that will form another plant. That is the purpose of fruits. They're literally plant babies covered in sugar for animals to disperse in the environment. So it's weird when you think about it, but that's what fruits are. Good. Any questions at all?
I had a question.
Yeah.
So, you know how you said cross-pollination is when the pollen from one flower goes to another's stigma? If it's like a different like species or variation of plant, does it still happen, or will it not work?
Very good question. Now, this is the thing about plants versus animals, etc., there's a difference between breeds and species, right? As you all know, we can combine two different dog breeds and get a hybrid, but dogs are all one species. If you were to get two different plant species, you cannot make an offspring. It is not possible, because the definition of a species are two organisms that can combine to make an offspring that is fertile. Those are the two criteria you need to be called a species, right? So if you get two different plant species, by definition, they cannot make an offspring. But if you get two different strains of a plant, if you get wheat from Canada and wheat from India, right, and you combine them, they're both the same species, so they will make an offspring. They're just different strains or breeds. Does that make sense?
Yeah, thank you.
Good question. That's selective breeding, right? So in wheat in Australia, we actually combine Canadian wheat, which is good for baking, etc., with wheat from India, which is drought and disease resistant. You got this hybrid which survives very well in Australia. Good. Okay, I know you all know what fruits look like, so it's just examples of seeds. But you can appreciate how seeds look so different based on who is going to be dispersing them. When it's animals, they're all colored and coated in glucose. When it's wind, you see dandelions, right, which you blow. And then there's others which are self-dispersed, which will just eject themselves onto the ground. Good. Now, this last part is vegetative propagation. Now, what did we say about pollination? At all? Is pollination sexual or asexual?
Sexual.
Very good. So vegetative propagation is the asexual form of reproduction of plants. Okay. Now, in vegetative propagation, just think about what the word means. Vegetative means like dormant, relating to a vegetative state, right? When you say something is vegetative, it's dormant or sleeping, etc. And propagation, which is continue. So vegetative propagation is used by plants, mainly plants, and it's a way of mitosis and rapidly creating new plant. But literally what happens is mitosis of a plant to form either a new shoot system or a root system. So as you know, the two parts of a plant from year 11 are the roots, and then everything above the soil is the shoot. Okay, and then there can be leaves and there can be flowers, etc. Okay, now there's different types of vegetative propagation. They have different names based on whether it's the shoot system that is undergoing mitosis to make another plant, or whether it's the root system which is growing underground and then from the bottom up making a new plant. Different plants do different things. Here's an example of spinifex grass, right? So you see this around Uluru, Central Australia. What do you notice? The plant is running along the surface of the soil. So, Sarah, is that the shoot system or is that the root system that's propagating? Any ideas? Logically think about it. It's above the soil. Would it be the shoot or the root system?
Did you say my name?
Yes, Sarah.
Oh, sorry. It's the root system.
Close. Since it's above the soil, we'd say it's the shoot system, right? Because the roots are underground. So if it was the root system, you wouldn't see anything. All you'd see is plants popping up side by side along a linear direction. Okay? But since you see it above the soil, it's the shoot system. And it looks like they're running, right? Use your imagination. It looks like the shoot system is running across the soil. So we call these runners. This is the one example I want you to have. Runners, spinifex grass. And I just want to preface this, guys. A lot of students get carried away trying to memorize examples of everything in biology. I can promise you in the HSC exam, they're not going to ask you about a specific species example for anything, except for disease. Disease, you must know examples, because otherwise you don't understand diseases. But I mean like weird plants and what they do, forget about memorizing them. Just understand, and then it will come up in an exam likely as an MCQ question, etc. The one thing I want you all to realize is after all the learning you'll go through with me at the end of the year when you sit your bio exam, and this is what every single batch of students say, we'll say the exam was so much application and not much memorization and rote learning. And the old syllabus used to be that way, which is why some teachers still teach like that. Just remember, all this stuff is very low yield to memorize. The key thing I want you to remember is pollination and vegetative propagation, sexual or asexual, and a very brief one-liner on what each is with terminology. Good. Okay, forget all of this stuff, right? So here you can see it's underground new plants forming. There's tubers, there's rhizomes. All of that, like I said, is very low yield. So we're going to move on. And let me ask you an exam question. One of the key things all of you should be doing from an early set point is exam questions. Now, I'm going to be writing your homework right now in the Teams channel, so all of you know what to do. Now, regarding homework and checking, guys, this is how it's going to work. The next lesson, I'm going to be asking random students to give me their answers to each of these questions, and I'll very briefly tell you what will be the marking criteria for each of these checkpoints. Okay? At the end of the 10 weeks, you will show me evidence of completion of the entire booklet so you can sit the exam. So the 10th week of this year, or this term, you'll all be sitting a Module 5 exam written by me. The exam is going to be at the level of your trial exams. It's going to be harder than your normal school exams, but if you pay attention, you put in the work in class, you'll smash it, right? 80% or so is a smash for that exam. So that's the structure of how we're going to be doing homework, etc. So we won't need to submit it because that's quite cumbersome, especially for online classes. But what you will need to do is make sure it's done. So if I ask you, you have an exam answer ready. Good. So for this week, you're going to be doing checkpoint C, and I'll be writing this as well. For homework for this week, for 22nd of the 10th, is going to be checkpoint C, which is on page 27. And I want you all to do checkpoint D as well, which is page 33. And pages 33 to 41. There's not many questions in 33, 41, just a couple. Yeah, so generally the homework is going to be whatever content we get up to, whatever page we finish the lesson on, all the questions between those pages, I want you to do it. Again, I'd highly recommend doing it now. Don't do it, you will have huge gaps in your content knowledge, okay? Good. I would just say go all the way up to about page 42. Okay, let me just submit that in the Teams channel. Good. All right, coming back to this, the exam question I'd like you all to do is going to be this one. This will teach you all a lot. So you have 2 minutes to plan this question. I know it's a 5-marker. You're not answering the question. All I want you to do is, instead of writing a full 5-mark exam answer, just tell me what your answer is and briefly why. So you can jot down a few points, but you have 2 minutes to read and think of an answer to the question. Good. All right, so in 2 minutes, I'm going to ask someone what their general answer to this. So, Atul and Alyssa, just regarding the question that I ask all the students, Any ideas on what you two guys want to do next year? As in, not next year, but when you get to university. Any general field that you're interested in, any jobs, etc.?
Medicine.
Okay, good. And Alyssa, what about you? Any ideas? Oh, I think our connection timed out. Cool. A small point I'm going to tell you guys, especially with uni courses, is please don't think about degree. Think about the job or the field you want to be in as an adult, and then work back to the degree you need to work in that job. Key example I see so many students make as a mistake is they pick medical science because, oh, it's high ATAR, it's probably going to be good and it's something to do with the medical field. If you do medical science, you'll end up $60,000 in HECS debt with no job security at all afterwards. The only jobs you can pursue is teaching. We have to do another degree or research, which is very, very strenuous and very competitive because there's not much funding in Australia for research. Versus if you're a doctor, you can do research on the side and you can do clinical roles, surgery, public health, and there's so much variety to it. And for that, you need a medical degree. Okay. So please don't think degree, think what job area do I want to be in, and then think what degree would help me best position myself for that. Good. Okay, let's go through this question, exam question 1. All right. So, Matthew, I'll get you to start us off. What do you think this organism can be based on what's been described? Let me help. Before we answer this question, let's understand what these fancy words mean, right? Inoculate and petri dish. A petri dish is this little container like this, and it's got jelly inside. Not the jelly that we eat, but agar jelly. Agar is a protein from seaweed. It's a very clear colored protein, and it's in this little plastic disc. We put a lid over that disc, and before we put the lid, we streak that surface with a long tube, a long rod, a metal rod that contains bacteria from a sample. It could be from toilet water or fresh water or a swab from a patient's nose or mouth, right? And then the bugs will start growing on the surface. And then we can look at it under a microscope. So that's what petri dishes are. And inoculating is a process of smearing the sample on that petri dish. Okay? Good. So, yeah, Matthew, continue. It's unicellular. So straight away, what are you thinking with unicellular? What are the options we have?
I think it's a eukaryotic cell.
Yes, why do you say that?
Because it has a Golgi body, which is membrane bound.
Good. And it's unicellular. So, Sarah, what are our options for a unicellular organism? What options do we have? It's hard because you need to go through the list.
No.
Yeah. Take it away.
Bacteria is multicellular, right?
Bacteria is unicellular.
Sorry, yeah. Bacteria.
Good. That's one option. Rithania, what's another one?
Fungi?
Good. Bacteria. Fungi. What other examples do we have at all?
Protist.
Protist. Very good. That's all we have because plants, animals, they're all uni. They're all multicellular, right? So these are only unicellular organisms. Very good. So he concludes, so it can't be bacteria because Matthew said it has to be eukaryotic and bacteria prokaryotic. We've got fungi and protists. And the student says it can only engage in asexual reproduction. Ed, what are your thoughts on that? Edward.
Well, it means can't be either because fungi and protists do both.
Fungi do both through spores. Do protists do both, Ed?
Oh, I should know.
Protists do only asexual, right? Because, Anne, can you just remind everyone, what are the two types of reproduction of protists?
Protists. So there was binary fusion and budding.
Very good, right? And with fungi, it's just there's budding and there's also spore formation. Very good. Now, final thing, right? A lot of students, me, everyone, going through Year 12 struggles with how do I get 5 out of 5 in an exam question? So let me briefly show you exam technique, which is what all of you should be working on through the rest of this year to do well. Because even if you know 100% of the content, you'll only get 80% in an exam if you don't know how to answer the questions. So we'll start with this. What is the most important word that will frame your answer to this question? One word, the most important one that will frame the structure of your answer.
Evaluate.
Very good, right? The question verb. All of you by now should have done what I told you last lesson, which is print the glossary or the NSW Science HSC verbs and stick it on your wall. So you get very familiar and used to seeing it. Now, what does evaluate mean? Now, I know all of you are going to say evaluate means make a judgment. But I want more, because you'll only get 1 mark for a judgment. Where do the remaining 4 marks come from, everyone? So let's go to Nathania. What does evaluate mean, Nathania?
To give more information to lead to the judgment.
Good, but do you agree the more information part is a little bit vague and you might accidentally brain dump because it's like, what do I do, right? Let me show you exactly what exam technique is. Evaluate means judgment and then pros and cons. And since this question is more than 4 marks, you should be defining key terms. Defining key terms. What's the key term we want to define here? There's one key term you want to define. What would it be? Someone tell me.
Would it be asexual?
Very good. What is asexual versus sexual reproduction, right? So in my answers, I just write definition. I don't give it a mark because it could be that it's not part of the marking guideline, right? But you want to be safe. So always include a definition when it's more than 4 marks. That's one part of exam technique. The next part is, since this is an evaluate question, I know straight away I need to say judgment, I need to say pros, I need to say cons, right? For pros for this statement, the main pro is protists, right? Because protists engage only in asexual reproduction. So my pro or points supporting this question are going to be about protists. Now, I have 4 marks left, you'll agree. It's a 5-mark question. I've got 1 mark for my judgment, which means I've got 2 marks for pros, 2 marks for cons. And now when I say the word protist, I need to first of all identify what they are, which is unicellular eukaryotic organisms. And then I need to briefly talk about their type of reproduction, which is binary fission and budding. And then I can say 2 marks or points against the statement. So first of all, I'm talking about fungi, so explaining what fungi are. And then 1 mark for what fungi engage in, which is budding and spore formation. And guess what? Now I've got 5 points that I'm going to say to make sure I get 4 marks for these questions. Good. All right. We'll end the lesson here because we've hit time. But does anyone have any other questions? Good. You're all doing really well. When you get your assessment tasks, please, first of all, you can send it to me on chat, but I'd actually like, if you could send it to me on chat, that'd be good. And I'll give you some feedback on how to approach the task. make sure whenever you send me assessment task, you don't just send me the notification. Okay, I need you to send me a very concise explanation on what you need from the task and specific questions that you have for me as well. And then I'll also give you general advice on top of that. And then you can send me work to quickly have a look at and skim, and I'll help you outside of class with that. Okay, but I need you all to make sure you give me a specific focus question, like, hey Prithvi, or hey sir, I have difficulty with what materials I need for my model or meiosis or DNA replication. I'm thinking of using toothpicks, XYZ. What do you think? And then I can give you advice on that. Okay? Good. Good luck. You're all doing really well. Keep it up, and I'll see you next week. Bye. See you guys.
Thank you.
Bye.